So let's start a new subject, that is biochemistry. In biochemistry, we will first learn about the protein and amino acid, then general biochemistry, then enzymology, then carbohydrates, then about the lipid metabolism, then vitamins, then molecular biology, bioenergetics. Okay, let's talk about the amino acid. So what is amino acid? So it is containing carbon, okay, then NH2 mandatory and COOH mandatory. Okay, now the other two groups will be changing. Okay, so if like there are 21 amino acid, okay, so every amino acid has different structure. Now the classification of amino acid. So classification of amino acid, it is based on side chain, it is based on metabolic fate, based on nutritional requirement. Okay, so if you can see, if this is the basic structure, NH2 and COOH is present in every amino acid. Okay, now H is there. Now this R can be replaced. So if there is H, then it is glycine. Okay, so if this R is changing in every amino acid, and it will form the new different amino acid. Okay, this is the basic structure. Are you getting my point? This is the basic structure, and this R will be changed in every amino acid. Okay, now classification of amino acid on the basis of side chain. So the mnemonic is CM will set alag guess for pretty well. So what is the meaning of this mnemonic? So you can see these CM has different class like CM, cysteine and methionine. So this is sulfur containing amino acid. Then will, what is will? Valine, isoleucine and leucine. So these will is branch containing amino acid. Then set, what is set? Serine, threonine, tyrosine. So it is hydroxy containing amino acid. Then alag, what is alag? ALA is for alanine, G is for glycine. This is simple amino acid. Okay, alag gas is for, G is for glutamic acid, aspartic acid. These are acidic amino acid. For you have to skip. Then pretty, what is pretty? PR is for phenylalanine, T is for tryptophan and TY is for tyrosine. So these are aromatic amino acids. So are you getting my point? What is the meaning of this mnemonic? So this one word constitute a different group of amino acid. Like CM is the sulfur containing, VIL is for branch containing, set is for hydroxy containing, alag is for sulfur containing. Okay, gas is for, these are the acidic amino acid. Okay, for you have to skip. Pretty is for aromatic amino acid, behel is for basic amino acid, B is for behel, B is for basic amino acid, and HAL is for histidine and arginine and lysine. Okay, so this one word constitute a different group of amino acid. Now histidine has imidazole group, arginine has guanidinium group, and tryptophan, so trypto has O, so it has a indole group. Okay, now amino acid on the basis of its structure. So you can see I have already told you this is glycine. So glycine has only H. This is permanent. You can see this NH2 is permanent, COOH is permanent, and this H is also there, and the R group will be changed. So if there is H, then it is glycine. If there is CH3, then it is alanine. If we have branched like CH3, CH and CH3, then it is valine. Okay, you can see this is branched. One more carbon is added, so it is leucine. Okay, so these are different type of amino acid based on their structure. You can see these are the aromatic amino acid. This is phenyl. This is structure is phenyl. Now this is alanine, so name is phenylalanine. If you put OH in phenylalanine, so this is tyrosine. Okay, this is indole group, so this is tryptophan. This is indole group, so it is tryptophan. Okay, this is phenylalanine. If you add OH, then it is tyrosine. Okay, this is proline, and you can see this is S, so it is sulfur containing amino acid. This is methionine. Okay, COOH, so this is COOH, this is permanent, this is permanent, H is permanent. So we have added one more COOH, so it is acidic amino acid. Okay, this is aspartic acid. Okay, now instead of COOH, so if you add CONH2, so it is aspargine amide. Okay, now amino acid and their structure, so amino acid could be polar or non-polar. So polar amino acid could be charged or uncharged. So charged is acidic. So what is acidic? We know aspartic acid and glutamic acid, and basic amino acid we know hull. So histidine, arginine and lysine. So these amino acid, are you getting my point? Like you can see this is COOH is permanent, and NH2 is permanent. So this is negative charge and this is positive charge. So negative and positive will cancel out each other. So if you add COOH extra, that will increase the charge on it. So aspartic acid has polar amino acid. Glutamic acid, if you increase one more COOH, so that is glutamic acid. So both have the polar. Okay, are you getting my point? Acidic and basic amino acid are polar because of extra COOH or extra NH2. Okay. Now, what is uncharged amino acid? So, those amino acid which does not have any charge, like hydroxyl containing amino acid, like cysteine, glycine, these does not have any charge. So, these are uncharged amino acid. Now, non-polar amino acid. So, non-polar amino acid are branched chain amino acid, aromatic amino acid, sulfur containing simple and amino acid. The mnemonic is BUSC. What is BUSC? Branch chain aromatic sulfur containing simple and amino. So, this is BUSC. Okay. So we have learned about amino acid on the basis of their structure, on the basis of side chain. Okay, now let's talk about amino acid based on the metabolic fate. So what is metabolic fate? Like, let's talk about the glycine. So what will glycine form if it is undergoes metabolism? Okay, what will phenylalanine form if it undergoes metabolism? Whether it is forming ketone or glucose or both. So the mnemonic is LL. So what is LL? Leucine and lysine. So LL is the ketogenic amino acid. Okay. Next is trypti. So what is trypti? TR is for tryptophan and P is for phenylalanine and T is for tyrosine. It is not tryptophan, it is tyrosine. I is for isoleucine. So these are glucogenic as well as ketogenic amino acid. Okay. So trypti is both tryptophan, phenylalanine, tyrosine and isoleucine. These both are glucogenic as well as ketogenic. Leucine and lysine are ketogenic only, okay, except these ketogenic as well as ketogenic and glucogenic. All other amino acid are glucogenic, okay. Now amino acid based on nutritional requirement. So what is the nutrition requirement? Like essential amino acid. So essential amino acid are those amino acid which is required by the body from outside, okay. So the mnemonic is Matt will fly. So what is Matt will fly? M is for methionine, T is for threonine, T is for tryptophan, V is for valine, I is for isoleucine, L is for leucine, fly is for PH is for phenylalanine, LY is for lysine. So these are essential amino acid. Essential amino acid are those which is required by the body from the outside. Okay. Now what is semi-essential? So semi-essential amino acid are histidine and arginine. Okay. So we have understood about the essential amino acid. Okay, we have understood about the semi-essential amino acid, that is all others except these are non-essential. Okay, now what are the derived amino acids? So we know 19 amino acid are the simple amino acid. Okay, they are naturally present. Okay, now if we modify the standard amino acid, that is derived amino acid. Okay, so 19 is 20. So like lysine, we know lysine is there. So if we add the OH, so that is hydroxylysine. So it is a derived amino acid. Okay, seleноcysteine. So it is a 21 amino acid. 22nd amino acid is pyrrolysine. So are you getting my point? If you modify the cysteine, so it is seleноcysteine. If you add the selenium compound, so codon UGA, stop codon is converted to the coding codon. Okay, pyrrolysine. Okay, so these are derived amino acid. Okay, what is derived amino acid? Derived amino acid are those amino acid which are derived from the standard amino acid. Like lysine is a standard amino acid. So if you add hydroxy compound, so it is a derived amino acid. Okay. Okay. Now what are the properties of amino acid? So amino acid absorbs UV light. It exhibits isomerism. Isomerism, why? Because it has asymmetric carbon. So asymmetric carbon, what is asymmetric carbon? Asymmetric carbon is that carbon which have 4 different valencies. We have learned in 11th class, the carbon which has different 4 attachment with that carbon, so that is known as asymmetric carbon. Okay, so like it has H or COOH, so and NH2, and the fourth is different, like except glycine, because glycine has H, so both have not, this is not asymmetric carbon. So if there is alanine, so you can see this carbon has 4 different valencies. that is COOH, that is NH2, then H, then CH3. So this is, it exhibit isomerism. So because it has asymmetric carbon. Okay. Now glycine did not, because it does not have chiral carbon. So because this C and H is present only. Okay. Okay. Now what is isoelectric point? So isoelectric point is average of ionization constant of ionizable group. Okay, that is isoelectric point. It is present in amino acid. Okay. So pH at which molecule has no electric charge, no mobility at the electric field. That is isoelectric point. Okay. Now, what are the fibrous protein? So fibrous protein is collagen. So, uh, so what is collagen? Collagen, it is most abundant protein present in the matrix. So you can see collagen is present in skin. It is present in cornea. Okay, highest density of collagen is present in cornea than skin. So what is the structure? So structure is triple helix, polyproline, alpha chain, quarter staggered. Okay, glycine is the major contributor. Okay, what is the synthesis? So synthesis is in rough endoplasmic reticulum. Okay, now you can see it has different types. So type 1 is present in bone. Defect in type 1 leads to osteogenesis imperfecta. Type 2 is present in cartilage. Type 3 is present in skin, lung, vascular system. Type 4 is present in basement membrane. Okay, so these are the different types of fibrous protein. Different type of collagen. So collagen type 1 is present in bone. So bone. Mnemonic is 1. Cartilage is 2. So this collagen type 1 is present in bone, type 2 cartilage, type 3 is in skin, lung and vascular system. Type 4 is in basement membrane. If there is defect, then it leads to Alport syndrome. Okay. Type 4 is present in basement membrane. Type 3 is in vascular. Now next is elastin. So what is elastin? So elastin protein is present in lungs, arterial blood vessels. Okay, so elastic recoil is there. Okay, keratin. So keratin is protein present in hair, nails. This is the keratin protein is present in hairs, nails. It is rich in cysteine. Okay, disulfide bond is there. More disulfide bonds, harder is the keratin. Okay, so disulfide bond is responsible for the keratin. Now fibrillin 1, it is largest glycoprotein. It is structural component of microfibrils. It is scaffold for the deposition of elastin. Okay. So now let's talk about the amino acid metabolism. So what is amino acid metabolism? It is the first reaction is transamination reaction. So what is transamination reaction? So normally we know there is an amino group. Amino group like this is C. This is C. This is NH2. This is COOH, and this is H, and this is R. R is changed in every amino acid. So we know there is a NH2 group. So this is amino group, and there is a keto acid. So what will happen? It form in amino acid, then along with amino acid plus keto acid, so it form the new amino acid and the keto group, keto acid. So you can see this is the example. This is alpha ketoglutarate. This is a keto acid. This is aspartate. Okay, this is aspartate, so it is amino acid. So amino acid plus keto acid, so it form a new amino acid. So what is the new amino acid? That is glutamate. And new keto acid, that is oxaloacetate. Okay, are you getting my point? So amino group from one amino acid is transferred to the keto acid. So it form new amino acid and new keto acid. So this is the transamination reaction. You can see this is the alpha ketoglutarate and alanine. This is alanine and this is alpha ketoglutarate. So they react and form the pyruvate and glutamate. So glutamate is a new amino acid and pyruvate is a keto acid. Okay. So this is the transamination reaction. So almost all amino acid undergoes transamination reaction except LAPAT. What is LAPAT? L is for lysine, P is for proline and T is for threonine. So this is LAPAT. LAPAT chalti hai na jab, so LAPAT does not undergo transamination reaction. So what is the coenzyme used? That is PLP, pyridoxal phosphate. Okay, B6 bitumen. Okay, this is the coenzyme used. Okay, amino group from alpha amino acid is getting concentrated as a glutamate. Okay, now the non-alpha amino acid that undergoes transamination is delta amino group of ornithine. Okay, that is transamination reaction. Now the ammonia, source of ammonia. So we know glutamate react with, glutamate is an amino acid, it react with NH3 and form the glutamine. So this is the reaction. This is glutamate, it react with NH3 and form the glutamine by the enzyme glutamine synthetase. And glutamine can convert it to the glutamate by the enzyme glutaminase. H2O converted to the NH3. So this is the reaction. This is how NH3 run into the body. NH3 is a gas, so it can't run without the reacting with any compound. So glutamate react with NH3 and form the glutamine, that is glutamine synthetase. Okay, glutamine is the transport form of ammonia from the most of the organs. Okay, now ammonia is transported in muscle in alanine form. Okay, so in muscle it run in the alanine form. In other body parts mostly it is glutamine. Okay, so it reaches liver in the form of glutamine and alanine. Glutamine can be converted to the glutamate we know, and glutamate to the alpha keto glutarate. So this is the reaction. This is the reaction. This is glutamate. It convert to the alpha keto glutarate. So the reaction is oxidative deamination. Okay, by the enzyme glutamate dehydrogenase. So this is the glutamate. Uh, this is the glutamate and the enzyme. It converted to the alpha ketoglutarate by the help of enzyme glutamate dehydrogenase. This reaction is oxidative deamination. Okay, so NH3 is released in this case. This is NH3 released, and glutamine is converted to the glutamate. So NH3 released here. So NH3 is released in both reaction. Glutamine is the transport form of NH3. So glutamine is converted to the glutamate and NH3 is released. Okay, glutamate again converted to the alpha ketoglutamate and NH3 is released. So glutamine is converted to the alpha ketoglutarate. Why? Glutamine is to then glutarate, then glutarate is to alpha ketoglutarate. So this is how NH3 is released. Now this NH3 is in liver. So where it goes, are you getting a point? From the other body parts, glutamine, from the other body parts, NH3 brings in the form of glutamine to the liver. Okay, and from the muscle in the form of alanine, it brings to the liver. Okay, now this NH3 has to go in urea cycle for the detoxification. So in liver, this released NH3 goes in urea cycle. Okay, this released NH3 goes in urea cycle. So what is urea cycle? First you have to understand it happens in two places. It is cytoplasm, it is mitochondria. So different reaction at different places. Okay, so first NH3 and CO2, it react, and because the enzyme is caromyl phosphate synthetase type 1, and 2 ATP is used. So 2 ATP is converted to 2 ADP and phosphate is released. Okay, so this is carbamyl phosphate, the first reaction of urea cycle. Okay, now carbamyl phosphate and ornithine, it form the citrulline by the help of enzyme ornithine transcarbamylase. Okay, citrulline is converted to the arginosuccinate. So if you want to remember the enzyme, so how will you remember? So remember the enzyme like arginosuccinate जो है तो वो बनता है पहले, तो उसके पहले वाला एंजाइम क्या होगा? आर्जिनोसक्सिनेट सिंथेटेस. उसके बाद वाला एंजाइम क्या होगा? आर्जिनोसक्सिनेट लाइज. ओके, पहले बन रहा है देन वो टूट रहा है. ओके, so first reaction is NH3 plus CO2. The enzyme is caromyl phosphate synthetase 1. The product form is caromyl phosphate, and 2 ATP is converted into the 2 ADP. Okay, now the caromyl phosphate react with ornithine and form the citrulline. Okay, citrulline is converted to the arginosuccinate. So the enzyme, because arginosuccinate is forming, and citrulline plus aspartate. So because arginosuccinate is forming, so the enzyme is arginosuccinate synthetase, and ATP is converted to the AMP, and 2-phosphate is released. Okay, arginosuccinate is converted to the arginine, and it releases the fumarate. The enzyme is arginosuccinate lyase. Okay, arginine is converted to the ornithine by the enzyme arginase. Okay, and H2O is used. So you can see ornithine is again going into the mitochondria and urea is released. Okay, so you can remember this cycle, and you have to in every cycle you have to remember the rate limiting step. So what is rate limiting step? So it is the crucial step. Rate limiting step is crucial step. If this step is not there, then the reaction will not proceed. Okay, now what are the component used? So you can see can is used. What is can? We know NH3 is used, CO2 is used, and aspartate is used. So that is can. Okay, and the rate limiting step is caromyl phosphate 1, and N-acetyl glutamate is the allostatic activator. Okay, now the energy at the step of CPS1 uses the 2 ATP. So you can see this is the 2 ATP. It will convert it to the 2 ADP. Okay, so we know that at the first step the reaction is utilizing 2 ATP. Okay, now the second reaction arginosuccinate synthesis, it utilized 2 phosphate. ATP is converted to the AMP, so 2 phosphate is used. So total 3 ATP and 4 phosphate used. How 3 ATP? Because in the first reaction 2 ATP is used, and in arginosuccinate synthesis 1 ATP is used. So 3 ATP and 4 phosphate is used. Now how will you remember this cycle? So the mnemonic is orange color cat always attract furious arginine. So the first reaction is ornithine, then caromyl phosphate. Okay, so ornithine and caromyl phosphate react to form the citrulline. Citrulline and aspartate react to form the arginosuccinate, and arginosuccinate is converted to the arginine and it releases the fumarate. So this is the mnemonic. Okay, now various defect and various enzyme defect. Okay, so the first enzyme is CPS1 defect. CPS1 is caromyl phosphate 1 defect. So caromyl phosphate 1 is defective. So the patient will present with hyperammonia. So CPS1, this is caromyl phosphate 1. If it is defective, so NH3 level will be increased obviously because it is the substrate. So because substrate is not there, so obviously, sorry, the enzyme is not there, so substrate will be increased. So that's why it is hyperammonia. Now hyperammonia type 2. So OTC2 defect. So OTC2 is ornithine transcaromylase. It is also used in pyrimidine metabolism. So uracil is increased. Okay. If we will study in pyrimidine metabolism, if OTC is defective, then there is hyperammonia type 2. We will learn in pyrimidine metabolism. Next is citrullinemia type 1. So obviously you can see this reaction. If there is arginosuccinate enzyme defect, so substrate will be increased. So citrulline is increased. So there is citrullinemia type 1. Now citrullinemia type 2 is citrin transporter defect. So what is citrin transporter? So you can see this is citrulline, and from mitochondria it has to come out to cystosol. So there is a transporter here. If this transporter is defective, the citrulline will not be able to come from the mitochondria to cystosol. So this is known as citrullinemia type 2. Okay. Obviously citrin is not able to come from the mitochondria to cystosol. So citrulline level will be increased. Next is arginosuccinic aciduria. So arginosuccinate level is increased because of defective arginosuccinate lyase enzyme. So the patient will present with arginosucciniduria. Okay. The patient will present with trichlorohexis, nodes are tufted here. Okay. Next is hyperarginemia. So hyperarginemia is because of arginase enzyme. So if arginase enzyme is defective, there is hyperarginemia because arginine is the substrate. The patient will present with progressive spastic dysplasia. This is the seizuring of gait. Okay. Now the third is HHS syndrome. So defective ornithine transporter. So where is the ornithine transporter? So you can see this is the ornithine, and it require ornithine from cystosol to go into the mitochondria. So here is the ornithine transporter. So if this is defective, so it leads to hyperammonia, obviously hyperornithemia and homocitrullinemia, because ornithine level is increased. So hyperornithemia, homocitrullinemia because citrulline level is increased, and hyperammonia because ammonia is free now. Ammonia because ornithine is not going inside the mitochondria, so ornithine level is increased. So ammonia is increased because ammonia is not able to form the ornithine. So it is a vicious cycle. So that's why it is a HHS syndrome. In ornithine transporter defect, all is increased. Ornithine is not able to go, so ornithine is increased. Ornithine के पहले क्या है? Citrulline, so citrulline is increased, and citrulline is formed from the ammonia, so ammonia is not is increased. Okay. Now the clinical features. Patient present with encephalopathy, respiratory alkalosis. Treatment, you have to provide the arginine, so it convert into the ornithine. Okay. So the next is aromatic amino acid. So aromatic amino acid is phenylalanine. So you can see this is the phenylalanine. You can see this is the structure of phenyl, and this is alanine attached. So the it is phenylalanine. If you attach the OH, then it is tyrosine. Okay, and this is tryptophan. Okay, this is tryptophan indole group. So it is tryptophan. Okay, now it has various reaction. Catabolic product it form and anabolic. So catabolic we know it form the ketogenic product and glucogenic product. Now, what is the reaction? So, the first reaction is phenylalanine, it's converted to the tyrosine. Then tyrosine is converted to the 4-hydroxyphenylpyruvate. Then homogenticate, then melilloacetate, then fumarate acetate, then fumarate and acetate. So, these are the reaction. These are the catabolic reaction. So, phenylalanine is converted to the ketone body and glucogenic. Sorry, this is glucone body and this is ketogenic body. Okay, so these are the catabolic reaction. And the various enzyme is used: phenylalanine hydroxylase, trans tyrosine aminotransferase, okay, homogenticate oxidase, melilloacetate, acetoacetate isomerase. Then last is fumarate acetoacetate hydrolase. Now, what is the mnemonic? Mnemonic is Precious tiger, hate hunting mighty fox. So Precious is phenylalanine, tiger is tyrosine, hate is 4-hydroxyphenylpyruvate, hunting is homogenticate, mighty is melilloacetoacetate, fox is fumarate acetoacetate. Okay. Now the various diseases. So various diseases in the enzyme defect. So if there is defect in phenylalanine hydroxylase, the disease is classical phenylketonuria. So we know the reaction phenylalanine is converted to the tyrosine with the help of tetrahydrobipentron, which converted it to dihydrobipentron. Okay, so this is the entire reaction. So if the phenylalanine hydroxylase is defective, it is classical. If this tetrahydrobipentron is defective, the enzyme used is dihydrobipentron reductase. It is the same reaction which is going parallel with this. Okay, so if that enzyme is defective, then it is type 2 and type 3 non-classic phenylketonuria. Okay. Now the third is non-classic phenylketonuria. It is type 4 and type 5. It is defect in formation of tetrahydrobipentron. Okay. Now next is alkaptonuria. So you can see if this enzyme is defective, homogenticate oxidase, the patient is present with acaptonuria. It is defect in homogenticate oxidase. Now the mnemonic next is fatty pH. So what is fatty pH? So type 1, type 2 and type 2 tyrosinemia. So how will you remember the enzyme defect in each condition? So the mnemonic is fatty pH. FAA is used in first enzyme. So that is fumarate acetoacetate enzyme defect. Second is fatty. So tyrosine transaminase defect, TT. And next is pH. So pH PP hydroxylase defect. So this is type 1, type 2 and type 3 tyrosinemia defect. Now various diseases. So phenylketonuria. So we have learned about it is the enzyme defect in phenylalanine hydroxylase. Okay. So phenylalanine does not convert to the tyrosine. It does not convert into the tyrosine because of this enzyme defect. So phenylalanine searches the other pathway. So other pathway searches and it form the phenyl acetate. So it form the phenyl acetate. So the mousy body odor. Okay. So phenylalanine did not convert it to the tyrosine. So the product formed by the tyrosine like melanin, catecholamine, thyroxine does not form. Obviously, phenylalanine is converted to the tyrosine by the help of enzyme phenylalanine hydroxylase. So this is the reaction, and tyrosine is converted to melanin, catecholamine, thyroxine. Okay, so if phenylalanine does not convert to the tyrosine, so it does not form the melanin, catecholamine, and thyroxine. So if it does not form the melanin, so the patient will present with hypopigmentation. If it does not form the catecholamine, so the patient will present with neurological deficit like agitation, hyperactivity, mental retardation, and the Guthrie test is used. It is the bacterial inhibition test. Not going in detail. I'm not going in detail. You need not to go in detail. Just remember this. It is a Guthrie test and ferric chloride test. It is positive. So because the phenylalanine level is increased, because the enzyme is defective, so substrate is increased. So what you have to do, you have to restrict phenylalanine. You have to give large neutral amino acid. Okay. So the next is alkaptonuria. So the enzyme defect is homogenticate oxidase. So in enzyme defect of homogenticate oxidase, so alkapton bodies accumulated in nose, pinna and sclera, and you have to do alkalization of urine. Okay. Ochronosis you will find. So what is ochronosis? It is alkapton bodies deposited in the intervertebral disc. Okay. ferric chloride test is positive, AgNO3 is positive. Okay, you will find in Gerard's tetrad, you will find the cap. So what is cap? C is for cystinuria, A is for alkaptonuria, A is for albinism, and P is for pentosuria. So you will find the cap in Gerard's tetrad. Now, how will you remember so many clinical features in alkaptonuria? So the mnemonic is itself alkaptonuria. Alka is for alkanization of neurine. P is for parrot-beak appearance of spine on X-ray. T is for it is a part of Gerard's tetrad. O is for ochronosis. N is for nitronison. It is used as a treatment. Okay. A is for AgNO3 test is positive. Okay. So this is the catabolic reaction of phenylalanine. Now, and the enzyme defect and the diseases. So now what is the anabolic fate of tyrosine? What is anabolic fate? So this is the reaction of anabolic fate. Phenylalanine converted to the tyrosine, which converted to the L-dopa, which converted to the dopamine, which converted to the norepinephrine. Okay, so these are the reaction. Phenylalanine converted to the L-tyrosine, then L-dopa, dopamine, norepinephrine, then epinephrine. Okay. So what is the mnemonic? For catecholamine biosynthesis, tired doctors don't need epinephrine. So what is tired? Tired is for tyrosine, then L-dopa, doctors is for L-dopa, don't is for dopa, need is for norepinephrine, epinephrine is obviously epinephrine. Okay, so we have learned about these various amino acids. Now let's talk about the glycine and serine. So glycine, glyoxylate is converted to the glycine with the help of enzyme glycine aminotransferase. Okay, so alanine is converted into the pyruvate. This is a transamination reaction, and the PLP is used in transamination reaction. We have understood, we have learned about it. Okay, so the catabolic product of glycine is pyruvate, and the anabolic product, so glycine is used in creatinine formation. It is used in heme formation. So heme formation we will learn in 2 or 3 lectures. In heme synthesis, succinyl choline react with glycine. Okay, so it form the heme. Okay, it is a component of glutathione. Okay, in purine it is C4, C5 and N5 of purine is glycine. Okay, serine can be converted to the glycine, threonine can be converted to the glycine, glyoxylate we know it is the transamination which is converted to the glycine. Okay, and THFA is converted to the N5 N10 methylene THFA by the use of glycine cleavage system. Okay, threonine is converted to the glycine by the threonine aldolase. Okay, serine metabolism, glycine is converted to the serine, glycine can be converted to the cysteine. Okay, we are not going in details, we are not going in reaction of all these, we don't have time, this is a rapid revision course. Now what is the use of serine? So serine uses is phosphatidyl serine, sphingosine and selenocysteine. These are the uses of serine. Okay, now the disorder of glycine and serine. So disorder of glycine, we know this is the reaction. So if this enzyme is defect in glyoxylate alanine transferase, if this enzyme is defective, this is the transamination reaction. So the patient will present with primary type 1 glyoxylate, the patient will present with hyperoxaluria, and the type 2 is glycerate dehydrogenase. So this is the primary hyperoxaluria. If the secondary is because this reaction requires the vitamin vitamin B6. So if vitamin B6 is defective, so the patient will present with hyperoxaluria and vitamin C toxicity. And so vitamin B6 deficiency and vitamin C toxicity both leads to secondary hyperoxaluria. Okay, next is sulfur containing vitamin. So methionine is converted to the S-adenosyl methionine, then it converted to the S-adenosyl homocysteine, then it converted to the homocysteine. Now homocysteine has 2 fate, it could be converted to the cystothioneine or it could be converted to the methionine. Okay, so homocysteine uses vitamin B12 and THFA for the regeneration of methionine. Okay, so if there is this reaction, homocysteine is converted to the methionine, and this reaction require vitamin B12 and THFA. So if vitamin B12 is defective, then the patient will present with homocysteinuria because homocysteine will not able to convert into the methionine. So homocysteine uses vitamin B12 and THFA for the regeneration of methionine. So if there is deficiency of vitamin B12 and THFA, then homocysteine level will be increased. So this is the remethylation reaction, and there is homocysteine can converted to the cystothioneine, that is the transsulfuration reaction. Okay, now there is a catabolic fate of methionine is glucose form the glucose body. Okay, homocysteine can also convert it to the cystathione, we know, and the enzyme used is vitamin B6. Okay, So vitamin B9 and B12 is used in conversion of homocysteine to methionine, and vitamin B6 is used in conversion of homocysteine to cystathionine. Now the mnemonic for methionine metabolism is my strong smart horse can race. So my is for methionine, strong is for acetinosyl methionine, smart is for acetinosyl cysteine, horse is for homocysteine, can is for cystathionine, and it's transsulfuration reaction, then cystathionine, race is for remethylation, methionine regeneration. Okay. Now the transmethylation reaction, norepinephrine converted to the epinephrine, epinephrine to the metanephrine, acetyl serotonin to the melatonin. Okay, decarboxylation reaction, lysine to the cadherin, ornithine to the putrescine. Okay, so these are the transmethylation reaction. Okay, just remember the name कि norepinephrine में CH3 जोड़ेंगे तो epinephrine बनेगा, epinephrine में CH3 जोड़ेंगे तो metanephrine. So these are the transmethylation reaction. Now various diseases related to the methionine metabolism. So first is Posthaus syndrome. So it is defect due to methionine transporter. Primary hypermethonemia. So methionine adenosyl transferase is defective. So this is the, so these all the reaction. So methionine is converted to the homocysteine. Sorry, there are various reaction of methionine is converted to the uh, methionine is converted to the as adenosyl methionine. So the enzyme is methionine adenosyl transferase. So if this enzyme is defective, the patient will present with primary methonemia. Okay. Next is classical homocysteinuria. So cystathionine beta synthetase is required to conversion of homocysteine to cystathionine. So if the enzyme is, so you can see this is the homocysteine and it requires cystathionine beta synthetase for convert to the cystathionine. It can convert to the methionine by the remethylation. Okay, so if this enzyme is defective, the patient will present with homocysteinuria. Okay, homocysteinuria. Okay, now what is the non-classical homocysteinuria? So if vitamin B12 and methyl THF is not present, so homocysteine will not be able to convert it to the methionine. Are you getting my point? Homocysteine can be converted to the methionine or it can be converted to the cystathionine. If it converted to the methionine, the reaction is transmethylation reaction and vitamin required is vitamin B9, that is folic acid, and vitamin B12. So if there is defect in vitamin B9 and B12, the patient will present with non-classical homocysteinuria, and that homocysteine can be converted to the cystathionine. The reaction is sulfuration reaction. Okay, and the enzyme and the vitamin required is vitamin B6. Okay, so that is classical homocysteinuria. Okay, now cystathione can be converted to the so cystathione can be converted to the cysteine by various reactions. Okay, this cystathione can be converted to the cysteine by various reaction by the help of enzyme cystathionase. So if this cystathionase is defective, the patient will present with cystinuria. Okay. Now, what is homocysteinuria? So it is due to defective cystathionine beta synthetase, that is vitamin B6. And homocysteine level is increased because homocysteine is not able to get converted. It is a substrate. Okay. Patient will present with mental retardation, ectopia lentis, thromboembolism, pectus carinatum, pectus excavatum, cyanide nitroproside test is positive. Okay. So how will you treat this patient? By giving the high dose of vitamin B6. Okay. And the non-classical homocysteinuria, how will you treat? It is due to defect in formation of N5-methyl THFA, methylcobalamin. We know, we have already discussed, it is due to vitamin B12 or vitamin B9 deficiency. So it is non-classical homocysteinuria. Okay, this is homocysteine. It has two reactions. It can be converted to the cystathionine or it can be converted to the methionine. So if this enzyme is defective, so the patient will present with classical homocysteinuria. If this enzyme is defective of remethylation, then it is non-classical homocysteinuria. Okay. Now this is cystathionuria. We have discussed that it is due to cystathionase enzyme defect. It is a we have already discussed. Now next is cystinosis. It is lysosomal storage defect. I am not going into detail of all this. Okay. Next is glutathione. What is glutathione? It is gamma glutamyl cystenyl glycine. So the glutathione full form is glutathione is made up of gamma glutamyl cystenyl glycine. Okay. Now the Garrett's tetrad, we know it is CAP. So CAP is for cystinuria, alkaptonuria, albinism, and pentosuria. And the excretion is cola. What is cola? C is for cysteine, O is for ornithine, L is for lysine, A is for arginine. Now tryptophan. So tryptophan has catabolic fate as well as anabolic fate. So in catabolic fate, it runs through the chironin cycle. So tryptophan is converted to the N-formyl chironin, and it undergoes multiple cycle, and finally it converted to the NAD, or it is the catabolic cycle. Okay. Now what is anabolic cycle? Now let's talk about the anabolic pathway. So anabolic pathway. So tryptophan is indole group, and it is used in formation of serotonin and melatonin. So the first is L-tryptophan, then it converts to the hydroxytryptophan, then it converts to the serotonin, then acetyl serotonin, then melatonin. So these are the reactions. Okay. So the mnemonic is the tri-sealing snack set mela. So these are the reaction which helps in formation of serotonin and melatonin. Okay. Now branch chain amino acid. So branch chain amino acid is BIL. What is BIL? Valine, isoleucine and leucine. So these are the first reaction. You can see this is the leucine, isoleucine and valine. Okay. It converts, the first enzyme here is branch chain keto acid decarboxylase. So and after multiple reactions, it convert into the acetoacetate. Okay. And here is glucogenic compound. So basically, what is the catabolic reaction of these branch chain amino acid? They ultimately form the glucogenic compound and ketogenic compound. Okay. The first enzyme which is used in these three amino acid is branch chain keto acid decarboxylase. So this is the enzyme which is first used. Okay. Sorry, the branch chain keto acid dehydrogenase. This is the enzyme. It has three enzymes. So what is the three enzymes? Branch chain keto acid decarboxylase, dihydrolipoyl transcyclase, and dihydrolipoyl dehydrogenase. These three enzymes constitute this enzyme. Okay, what are the coenzymes? So what are the coenzymes is CoA, TPP, lipoamide, FAD and NAD. These coenzymes are the part of this multi-chain enzyme. Okay. Now you need not to learn all these reactions. Just remember it is a glucogenic and ketogenic compound. We know already. Okay. Now the first enzyme, which is the branch chain keto acid dehydrogenase, if this enzyme is defective, the patient will present with maple serum urine disease. Okay. So what are the clinical features in maple serum urine disease? Patient will present with feeding difficulty, failure to thrive, lethargy. Okay. Okay. Now the various test is dinitrophenyl hydrogen test. It is positive. Okay. And the other disease is isovaleric aciduria, defective isovaleric acid enzyme defect. So that is isovaleric aciduria. Okay. Now histidine. So histidine is converted to histamine. We need not to know the entire sequence. Histidine form the histamine. Okay. And piglu is excreted in urine because of folate deficiency. Okay. So just remember these two things. Need not to go in detail. Just remember histidine form histamine, and piglu is excreted in urine because of folate deficiency. Just remember those two things of histidine. No need to remember the entire reaction. Okay. Now I will tell you the histidine after so many reactions form the histamine. Okay. And histidine in these reaction, because of folic acid deficiency, if there is no any folic acid is there, so it form the piglu. Piglu is excreted in urine. Okay. Need not to remember the entire name of piglu. Okay. They will give you in a question. Okay. Now next amino acid is arginine. So arginine form the nitric oxide, creatine and urea. Arginine form these three compounds. Okay. Now various disorder and the smell. So glutaric acidemia, it is sweaty feet smell. Oxyuria, swimming pool. Isovaleric acidemia, sweaty feet smell. Maple syrup urine disease, maple syrup smell. Okay. So these are the smell of these diseases. They can be asked in exam. Osthouse urine disease, boiled cabbage. Phenylketonuria, mousy smell. Tyrosinemia, boiled cabbage smell. Okay. So these are the smell of particular diseases. Okay. And these are the smells, and these are the particular diseases. Okay.